Afternoon, everyone. And I'm Mostafa ElShahed, professor from the Microbiology Department of Oklahoma State University. I'm a microbial ecologist. We do a lot of culture-independent characterization of microbial communities, how to go to a specific environment and identify the different types of microorganisms that are present in it without the need for culture. It started by a scientist by the name of Carl Woese. Carl Woese, his idea was we can sequence a specific gene within a microorganism, and the degree of relatedness of different types of microorganisms to each other is a function of how close this sequence is. Which gene did he choose for that? He chose 16S ribosomal RNA gene. He used to work in the University of Illinois in the 1970s, and they were very good in culturing a lot of microorganisms, aerobic and anaerobic. They have a large collection of microorganisms. So he borrowed all of these microorganisms from his colleagues, extracted the DNA through very crude methods, sequenced the 16S ribosomal RNA gene, and start looking into different relationships that are present between different microorganisms. Why did he choose a 16S ribosomal RNA gene? If you want to classify different microorganisms, you need to use a specific molecule and biomarker that is, number one, universally distributed. It's present in every single microorganism. It has to be functionally homologous, doing the same function in different microorganisms. It has to evolve at a suitable rate, not too slow, not too fast. And for technical reasons, it has to have a suitable size. It will have enough information for you to classify different microorganisms. Ribosomal RNA molecules are present in every cell because ribosomes are present in every cell. So these are the genes that encode the ribosomal RNA fraction, the ribosomal RNA part of the ribosome. And there's different molecules. The 23S is too large, 2.7 kb in bacteria. 16S is 1.5 to 1.6 kb, so it's a very good size. The 5S is 120 to 150 a base pair, so it's quite small. So he did that. And after he did that, he published a very famous paper in PNAS in 1977. He said to the whole world, hey, I compared all the sequences that I can find of the 16S ribosomal RNA, and I classify all different types of life that we have into bacteria, archaea, and eukaryotes. We call these three kingdoms or three domains of different lives. Eukaryotes, we all know what they are: large microorganisms that have a nucleus with a nuclear membrane. Many of them, animals and plants, are macroorganisms, but there's many micro eukaryotes like fungi and ciliates and so on. Bacteria and archaea are unicellular microorganisms that don't have a nucleus. Bacteria are most of the bacteria that you know, the bacillus and the E. coli and the Pseudomonas and all these different microorganisms. Archaea are more of a collection of less known microorganisms. Many of the original ones are extremophiles, but some new ones that we discovered are not extremophiles as well. And afterwards, Carl Woese continued working with that. He essentially asked everyone to sequence the 16S of their microorganisms, send him the 16S of different types of, send them DNA and cultures of different types of microorganisms. And eventually he published a paper in 1987, in which is really the basis of what we call phylogeny of microorganisms based on the 16S ribosomal RNA gene. He said to the whole world, look, I looked at all microorganisms that I can find, and I would classify them into 12 different phyla at this time. They call it the tree of life for bacteria: Acophax, Dermatoga, Greenland sulfur. Some of them were known to be a coherent group of microorganisms by themselves before that, and some of them are not. But essentially, this tree is the basis of what we call the 16S ribosomal RNA-based phylogeny. This is all good. This is all have been done with pure cultures. Until a scientist by the name of Norman Pace in the very early 1980s came up with a really simple, brilliant idea, which is the basis of all of the culture independent surveys that the whole world is doing right now. He said, well, wait a minute. If you look at this approach that Carl Woese is using, he is getting the DNA from a pure culture, sequencing the 16S ribosomal RNA gene from this pure culture. What if, instead of starting with pure culture, let's start with an environmental sample, a gram of soil, a milliliter of lake water, a fecal sample, any type of sample that we know that contains microorganisms. Let's extract DNA from this environmental sample. So the DNA that you get is a mix representative of all the genomes of the microorganisms that are present in the sample. Then you amplify the 16S ribosomal RNA gene using PCR with primers that you think are universal 16S primers, primers which would theoretically amplify the 16S ribosomal RNA gene of every microorganism in your sample. And there's many of these primers, and we continue to refine them and add redundancies to them to ensure that they're truly universal primer that would amplify all the 16S ribosomal RNA gene from all microorganisms. So now you have a PCR product. If you look at this PCR product in a gel, it's one single band, but it's really billions of thousands of different 16S ribosomal RNA sequences. And every single molecule in this band could theoretically, you could think of it as representing a single cell, a single microorganism that's present in your sample. So if you sequence the 16S product as much sequencing as you could sequence 100 molecules, you can sequence 1,000, 10,000, 1,000,000 molecules of this 16S ribosomal RNA PCR product. Every sequence you get is theoretically representative of a ribosome that came from a single cell that are present there. And the more similar sequences, so if half of your sequences are similar, you could imply that half of the microorganisms that are present in the sample belong to one simple species. And you can do a lot of analysis on that, but the basic idea is the relative proportion of the different types of sequences that you have are representative of the relative amount of microorganisms or cells in your environmental sample. The beauty of this approach is the DNA will be extracted and will be amplified of any cell, regardless of its preferred growth conditions, regardless of how easy or how difficult you can culture it. So a big problem in culturing surveys is you culture things that might be present in very low numbers, that might not be important for the ecological functioning of the system, but they just like to grow on the media that you give. When you do this type of analysis, you're amplifying what's closer to the truth in terms of relative amounts of microorganisms that are present in your sample, and you are amplifying cultured organisms that we know exist, but uncultured microorganisms as well. So this approach has started by Norm Pace, this very early paper by some of his students, and many of his students, each one of them went to a different environment and became the expert in culture independent surveys in this environment. So in soil, in the ocean, in many different environments. And collectively, when you think about thousands and thousands of studies of culture independent surveys, it not only tells us the microbial community in these environments, but it adds to our knowledge of different lineages, different groups of microorganisms that are parts of the chain. I had the pleasure in 2001 to go and visit the lab of Norman Pace in person. At this time, he was very active. He was working in so many different projects using this culture-independent approach for characterizing the community. I took samples from a place that I worked with called Zodelton Spring in Oklahoma. We worked on these samples and we did the whole phylogenetic analysis. I learned how to do it from him, and we published a lot of good stuff from this work. So if you remember, in 1987, we thought that the whole microorganism, the whole bacterial domain, could be encompassed in 12 different phyla. But after scientists have worked a lot and applied this culture-independent analysis in many different environments, by 2006, now we have a much bigger tree. But the whole idea here, the whole beauty of this approach for culture-independent analysis is you can go to a specific environment, you can go through specific procedures, and the results would tell you what type of microorganisms are present in this environment. And it has a huge impact of our understanding of the scope of diversity, as I said. So up to the 1990s, we didn't know that a group of microorganisms in soil called acidobacteria exist. Now we know 30 to 50% of the cells in soil belong to this group. We didn't know that the ocean, the pelagic, the open ocean, for example, 10 to 20 to 30% of the cells that are present belong to the group that nobody knew existed called SAR11. And you think of how vast the ocean is, how vast soil is, then really we were missing a lot of microorganisms. Sequencing technology is now much easier, so you can do a lot of these studies. You can get a lot of sequences per sample. You can do a lot of samples for a very, relatively speaking, cheap price, and that helped a lot in democratization of this type.